Hormones influence target cells by binding to specific receptors. The hormone-receptor interaction initiates the process of transforming the chemical message into the response of the cell. Your goals for learning are to learn about hormone receptors, to describe common second messenger systems and transcription factors, to review insulin, to understand that hormones induce changes in cellular metabolism. Here's what you need to know: that changing the shape of a molecule can activate or inactivate it. The terminology of carbohydrate, lipid, and protein metabolism. The difference between absorptive and post-absorptive states of the body. To see definitions of terms, click the bold red words. A target cell responds to a hormone with a sequence of events. Drag the hormone to the receptor to observe the events and response of the target cell. Receptor activation by the input signal, the hormone. Conversion of the input signal to a biochemical change in the cell. Conversions occur through second messengers and gene expression. Response of the cell. Examples of cellular response include contraction, secretion, transport, and synthesis or breakdown of storage molecules. We will study receptors here and conversion mechanisms and cellular responses later in this topic. Click Continue to proceed. Receptors are complex proteins, each specific for a particular hormone that binds with a high affinity. Hormones only affect target cells, cells that have specific receptors for them. For ease of viewing, receptors and hormones are drawn as simple shapes. Click the enlarged receptor to continue. Receptors are located in the plasma membrane or in the interior of the cell. Water-soluble hormones cannot diffuse into cells. They bind to receptors in the plasma membrane. Drag the water-soluble hormones to their receptors. Lipid-soluble hormones diffuse into cells where they bind with intracellular receptors. Drag the lipid-soluble hormones to their receptors. Recent evidence indicates that there are also receptors in the plasma membrane for lipid-soluble hormones, and that these receptors may induce more rapid responses of the target cell than intracellular receptors. Target cells have receptors for multiple hormones, and each hormone exerts a different action on the cell. Also, a given hormone may act on the same cell through different receptors, each inducing a different action of the cell. Not all cells possess receptors for all hormones. The number of receptors present in a cell may vary depending on the amount of hormone present. Prolonged high hormone levels may decrease the number of receptors and reduce the response in target cells, a process called downregulation. Continuous low levels of a hormone or another stimulating signal may increase the number of receptors and the sensitivity of the target cell, a process called upregulation. For example, hypersecretion of thyroid hormone upregulates receptors for epinephrine. This phenomenon leads to the increased heart rate and cardiac output seen in patients with hyperthyroidism. Click a thyroid hormone to see how hypersecretion upregulates receptors for epinephrine. Receptors are dynamic proteins, and like many other cellular proteins, they are recycled and replaced repeatedly during the life of a cell. The water-soluble hormones, peptides, and catecholamines bind to plasma membrane receptors on the outside surface of the cell. There are several categories of plasma membrane receptors, including receptors that activate G proteins and receptors with protein kinase activity. We will examine the most common G protein mechanisms and the protein kinases called tyrosine kinases that phosphorylate tyrosine. We will look at each kind of receptor, beginning with the receptors that activate G proteins. In the inactivated state, guanosine diphosphate, GDP, is bound to the G protein. Drag the hormone to the binding site on the receptor to observe its action. When a hormone binds to the receptor changes shape and activates the G protein. It releases GDP and binds guanosine triphosphate, GTP, a high-energy molecule. Part of the activated G protein travels in the membrane and activates an enzyme called adenylate cyclase. Adenylate cyclase catalyzes the conversion of adenosine triphosphate, ATP, to cyclic adenosine monophosphate. Cyclic AMP is a second messenger. The hormone is the first messenger. Cyclic AMP activates the intracellular enzyme protein kinase A, which can phosphorylate many other proteins, activating some and inhibiting others. This mechanism can induce many different effects within a single cell and different actions in different cell types. This second messenger system can also phosphorylate ion channels and alter their activity. The response of the cell ends when cyclic AMP is degraded to AMP in a reaction catalyzed by the enzyme phosphodiesterase. While active, G proteins keep stoking the pathway to phosphorylation of cellular proteins so that huge numbers of them are produced. This phenomenon, called amplification, greatly increases the effect that a single molecule of a hormone can have on a cell. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. G proteins, like the one we just described, that stimulate production of cyclic AMP are known as stimulatory G proteins. Inhibitory G proteins inhibit production of cyclic AMP and the subsequent response of the cell. Another type of G protein activates an intracellular messenger system that uses diacylglycerol and inositol triphosphate as second messengers. Drag the hormone to the binding site on the receptor to observe its activity. When a hormone binds to the receptor changes shape and activates the G protein. It releases GDP and binds GTP, a high-energy molecule. Part of the activated G protein travels in the membrane and activates an enzyme called phospholipase C. Phospholipase C causes a membrane phospholipid called phosphatidylinositol biphosphate to cleave into diacylglycerol and inositol triphosphate. Each functions as a second messenger. Diacylglycerol remains in the membrane where it activates protein kinase C, and inositol triphosphate enters the cytosol. The activated protein kinase C phosphorylates other proteins, causing responses of the cell. Inositol triphosphate prompts the endoplasmic reticulum to release calcium ions. Calcium ions also behave like second messengers. They can act directly on proteins or bind to the protein calmodulin that can act on proteins to cause the response of the cell. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. Drag each hormone to a binding site on the receptor to observe its activity. When a hormone binds to a tyrosine kinase receptor, the receptor joins with a neighboring receptor and changes shape. Each receptor phosphorylates tyrosine groups in its neighbor. The process is called autophosphorylation because the receptors are of the same type. The phosphorylated tyrosine groups provide docking sites where relay proteins bind. They initiate a series of protein phosphorylations that lead to the response of the cell. Receptors with tyrosine kinase activity often bind hormones like insulin and other growth factors that affect cell growth and differentiation. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. On this page, we will study mechanisms used by epinephrine and insulin to induce cellular responses. Nearly all body cells express receptors for epinephrine and norepinephrine called adrenergic receptors. There are two families of adrenergic receptors, alpha and beta, with each member of the family designated by a number. All adrenergic receptors bind both epinephrine and norepinephrine, although with different affinities. All adrenergic receptors activate G proteins. Beta receptors are coupled to adenylate cyclase by stimulatory G proteins that increase cyclic AMP activity. Alpha-2 receptors are coupled to adenylate cyclase by inhibitory G proteins that decrease cyclic AMP activity, often blocking the increases induced by other agents. Alpha-1 receptors act via the IP3-DAG and calcium second messenger system. Some cellular responses to these catecholamines are listed beneath the appropriate receptors. Blood flow is redirected to working muscles where increased cardiac output delivers increased fuel and oxygen. Fuel molecules are released from stores and made available for use by working muscles. The airways dilate to maximize gas exchange. Note that all of these effects support the fight or flight response. Insulin acts primarily on liver, adipose tissue, skeletal muscle, and cardiac muscle by binding to tyrosine kinase receptors. Each tissue responds differently, and each has multiple responses. Some of the cellular responses to insulin are listed beneath the receptors. Glucose uptake increases in all three major tissue types. Insulin stimulates glycogen synthesis in muscle and liver cells and triglyceride synthesis in adipose and liver cells. Amino acid uptake and protein synthesis are stimulated in muscle. Note that these effects support the overall goal of promoting storage of fuel molecules. The lipid-soluble hormones, steroids, and thyroid hormone bind to receptors typically located inside cells. Most of these intracellular receptors are located in the nucleus. Each receptor has two binding sites. One site binds the hormone, One site binds to DNA. In the absence of hormone, the receptor is bound to a chaperone protein that masks the DNA binding site and probably also acts to stabilize the protein. The receptors are not active until a hormone binds to them. Drag the hormone to the binding site on the receptor to observe its activity. When a hormone binds to a receptor, the receptor changes shape. This activates the receptor and causes the chaperone to disassociate from it. If it is not already in the nucleus, the hormone-receptor complex enters the nucleus. Hormone-receptor complexes act as transcription factors. They bind to specific regions of DNA called hormone response elements. Typically, the binding results in activation of or turning on a particular gene. When a gene is activated, messenger RNA, mRNA, is produced and enters the cytosol where it is transcribed to form proteins. Typically, the proteins that are synthesized are enzymes that catalyze biochemical reactions and cause the response of the cell. The properties of transcription factors are far more complex than the description above. Transcription factors can regulate more than one gene, and they can also decrease or turn off the activity of genes. Transcription factors for different hormones can bind to the same hormone response elements, although each transcription factor activates a specific group of genes. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. On this page, we will study the mechanisms used by cortisol and thyroid hormone to induce cellular responses. The action of cortisol follows the typical pattern for steroid hormones. It binds to glucocorticoid receptors that are present in nearly all cells. Some of the many cellular responses to cortisol are listed. Cortisol acts to maintain high levels of fuel molecules, maintain blood pressure, and modulate body defenses, all necessary for the stress response. We will study the stress response in Topic 5. The thyroid hormones, T3 and T4, enter cells by diffusion, and to some extent by carrier-mediated transport. Once inside, T4 is converted to T3, the active form of the hormone. Receptors for thyroid hormones differ from steroid hormone receptors. They are bound to the response elements of the DNA. T3 binds to the receptor, causing gene activation or eliminating the inhibition that was present, thus allowing genes to be expressed. Small amounts of T4 may also bind to the receptors. Receptors for thyroid hormones are also found in the cytosol and mitochondria, but their function is poorly understood. Nearly all body cells have thyroid hormone receptors and require thyroid hormone for normal function. Some of the many effects of thyroid hormone are listed. Thyroid hormone is the main regulator of metabolic rate in the body. Here's a summary of what we've covered. Hormone receptors are proteins located in the plasma membrane or the interior of the cell. Water-soluble hormones bind to plasma membrane receptors, and lipid-soluble hormones typically bind to intracellular receptors. Receptor activation engages cellular machinery that leads to the response of the cell. The effects of a single molecule of hormone are amplified by the cellular machinery. All about insulin, diabetes mellitus, and the causes and consequences of hypersecretion of insulin.